Great. Hello, everybody. Again, welcome to the webinar today for Bridging the Gap between Flow Cytometry and Results with FCS Express. Today's presentation is an overview of analysis efficiencies and capabilities of FCS Express. And the presentation today is really designed for researchers and scientists, folks in the flow cytometry field who have really little or no previous experience with FCS Express, and especially those who are using other flow cytometry data analysis tools for their software currently. And today we're going to be kind of going through how FCS Express works, kind of overall workflows. We're going to give you some examples of some of the tools and efficiencies that you can realize with FCS Express. Again, my name is Sean Burke. I'm the president at De Novo Software, and I'm really excited to be speaking to everybody here. So again, thanks for coming along. And I always like to start this talk or this presentation with, what are some of the biggest challenges that you have with flow cytometry data analysis right now? And this is a great opportunity. If you could type in the question in the answers box, it would be really great to hear from you. Where do you have challenges in your current software tools? We realize that a number of folks who are coming from other software packages, regardless of what they might be, have challenges around presenting data. They have challenges around managing statistics. They do a lot of copying and pasting. People are putting little white text boxes over access labels when they get out to Photoshop and PowerPoint. So again, if anybody wants to volunteer in the Q&A, I'm kind of taking an eye over there to see if anybody has any gripes or challenges that they'd like to put in here. Oh, great. I see, there's one coming in about exporting graphs with statistics. So that kind of falls into report generation, getting the final result. I know lots of folks have trouble in other software tools for instrument compatibility and managing gates. And I see a familiar one that just popped in there, managing very large data sets, right? Um, so these are again, oh, batch. I see batch processing coming through. So yeah, so some really great points coming through from the crowd here. And again, when we work in FCS Express, the idea is to overcome these challenges and overcome them in one place, right? And that's kind of where we're at with why use FCS Express. Because again, I know there's a lot of software tools on the market. There's a lot of things that you might be embedded in using right now because your facility, your institution, your PI, somebody's asking you to use those tools instead of FCS Express. But when it comes to our software, we like to say that we pull all of these kind of tools together. Things like cell cycle modeling, proliferation, phenotyping. We build into FCS Express and make that easy. The entire Microsoft PowerPoint suite of tools and exporting and moving between, working in a PowerPoint like interface, working with Excel like spreadsheets, having the ability to, export to PDF, direct exports to GraphPad Prism, Word, and we're going to work with, really any sort of cytometry. So if you're doing conventional, spectral, mass, imaging, we're going to pull those tools in. And if you're working with high dimensional data reductions, or, if you want to work with Python or R, or you want to use our native tools for high dimensional data reduction, we include all of that in FCS Express. Now, the other thing that kind of makes FCS Express special is that we have kind of two or three flavors of the software. We have the FCS Express RUO, Research Use Only Edition. We also have the FCS Express IVD Clinical Edition. And we have something that kind of works between them that we call Validation Ready. And I'm going to come in and talk more about Validation Ready. But essentially, with our clinical edition, the product is listed with FDA for IVD use. Our RUO product can be used for anything. And one of the really nice advantages of working with FCS Express is if you're working in research, you need to move things through to a quality regulated environment up until, a kind of clinical edition for either patient diagnosis or clinical trials or cell and gene therapy type work. FCS Express scales quickly and easily between all of those. Now, I mentioned validation ready, and you might be wondering, what does that mean? And who's that designed for? Is that me, right? Do I fall into that bucket? Because I know we have a lot of folks here from university. We have folks here from major biopharma companies on the call. We have folks here from kind of small and medium business laboratories. And essentially, the validation ready package is assembled for researchers who kind of work in cell and gene therapy space. It's really convenient to move between RUO and IVD with some sort of compliancy in between. Really, anybody working in a regulated environment. So, if you're not in cell and gene therapy, we have lots of people working in FCS Express who are just working in facilities where they need or they're considering some sort of GXP compliancy, including 21 CFR Part 11 compliancy. And the way that we help you get to compliancy with FCS Express is through a set of features that are integrated within the software. Our validation ready package is it's a self-hosted network license. It has fully definable security groups and permissions. So anything that you're doing in your analysis can be locked down, allowed, or disallowed for any level of user. It includes electronic signatures, full step-by-step logging and audit trails for every person on the system, as well as system-level audit trails. And again, these things can be enabled for the RUO or for the IVD, and we can take it a little bit further as well through additional available kind of features and tools like LDAP, single sign-on. We have an IQ/OQ program that's available. We have customized LIMS, LAS, ELN integrations with the software. So, we have a really, great background working in this space for a large number or many, many years, and you're going to kind of see a little bit more about that. And again, last thing on validation ready is that, some of the key components of this are that it can be integrated into new or existing workflows. So, if you're brand new to FCS Express, we can help get you supported. If you've been a long time user of FCS Express and you think this is right for you, you think it's right for, what your QC and science team needs to do, you can be helped to move into that. And the validation really leads to reliable and repeatable results in R&D. Um, and FCS Express with things like our IQ/OQ program and using the validation ready functionality really helps you ensure that you're working in a predictable, reliable manner, really enabling data integrity and traceability. And again, that's scalability, right? And that scalability can mean just bringing on additional users, or it could mean, you have a biotech site that's in, say New Jersey, and you need to scale that out to other countries and other locations around the country. We can do that. And FCS Express has a lot of experience working between IT teams, QC teams, science teams, and business teams. All of these people, really help these things come together. So that being said, I won't talk too much more about validation ready. I'm going to get into the software a little bit. But before I do, again, I want to outline some other key features of FCS Express. And one of those is support, because we know that if you're coming from other software tools, if you're new to flow cytometry, if you're new to FCS Express, you need support, you need expertise, you need people to help you kind of transition from what you were using or move into something new. Our company has folks, application specialists, experts kind of based throughout the world. Our headquarters is in downtown Pasadena, California. I work out of New Jersey. We have folks working out of the UK, Italy, all throughout the world. And it's really important because if you give us a call or you write us an email, say at 2 in the morning Eastern time, a lot of other software packages and tools, they're not going to get back to you very quickly. We often try and get back to people within an hour or two, never more than 24 hours. And if you write us an email at 2 in the morning East Coast time, we know you're not working 9 to 5. Well, we have our staff over in the UK, over in Italy that can help pick up and help move you across the time zones and keep you off and running. And as a little bit of background again about FCS Express, we've been around since 1998. If you haven't heard of us or you haven't used us yet, this is kind of important to know that we've been around. We've been working in this field for a very long time. When it comes to our clinical edition, it was first released in 2004, so over 20 years of experience, working in that sort of industry and that sort of market. We've gone through many iterations of FCS Express, introducing image cytometry functionality, introducing spectral capabilities, really keeping up to date. with everything that there is involved with flow cytometry in the market. In 2021, a company named Dotmatics, which you may have heard of because they own GraphPad Prism, Snapgene, Genius, some other major scientific software tools, purchased De Novo software. We've kind of gone through some iterations of that. And more recently, Dotmatics was acquired by Siemens. And we're very excited about, we've always been excited about working with Dotmatics and now being part of Siemens because of the opportunities it's going to afford us to give back to our customers to keep improving the software, keep moving forward, keep being state of the art and cutting edge with what we do. Now, if you, again, haven't heard of FCS Express and you're not sure if people in your institution or facility are using it, rest assured, FCS Express is being used all the time. As of a quick Google Scholar search earlier this week, there's over 15,000 and growing scientific publications that cite FCS Express as the flow cytometry software analysis package being used. Again, this is kind of a quick and dirty Google search, so I'm sure the number is actually, much higher than this. But just rest assured, people are publishing, people are using, people in your facility that you may not even know are using the software to, its full and efficient capabilities. And before we jump into FCS Express, so we can take a look at some of these efficiencies and functionality, I always think it's important to discuss that FCS Express runs on a Mac or a PC, and it's interchangeable. You can send data from your Mac over to your PC and back and forth. We're going to read data from every cytometer on the market, including specialized data reading for things like Cytec and Sony and Thermo instruments, image cytometry. So if you're working with Amnis, if you're working, with the Cytec imaging cytometers, if you're working with Thermo Cytpix imaging cytometers, other tools, we're going to be able to handle that. There's no limits to parameters or number of events in the data. So I know somebody had a question or a comment about, loading big data. Yeah, we can handle that. And we can handle that on your local computer because of the way that FCS Express have been developed to handle large data sets. And really importantly, compensation, we're not going to talk a lot about it today, but rest assured, we've been doing this for over 25 years. We've seen and worked with every sort of compensation and every sort of unmixing that you can imagine. We have wizards and tools that are built up within the software to help you succeed and easily proceed with any sort of compensation that you want. We also have the complete set of descriptive statistics. I know managing statistics was something that was in here as well. So working with things like MFI, percent gated, standard deviations, robust standard deviations, of course, that's all here. And all of the commonly used plot types, dot plots, density plots, contours, right? They're all here. And if you're coming from another software package and you don't see the plot type that you want, rest assured it's here, right? It's probably here, and we may call it something else, right? So if you're looking for something maybe called a zebra plot from another piece of software, we don't call it a zebra plot, but we call it a contour plot with black and white stripes, right? And if you've seen something published where there's a density overlay on one of your color dot plots or something like that, we have the ability to work with that. And our support team is always excited to hear from customers asking about how do I generate these figures that I saw published, and we can oftentimes direct you with minimal intervention to help you get off and running there. So I think, again, one of the big differences when we talk about FCS Express versus other flow cytometry software on the market is that a lot of other software tools, when you think about it, you're really just getting kind of basic plots and basic statistics that you're then having to copy and paste and move out to Prism and move out to Excel, having to reorder columns and rows of data. You're having to use tools like Photoshop and PowerPoint and R all to get a final result. So there's a lot of steps you're going through before you get that final result. And what we're going to show you in FCS Express today is that we're fully integrated for analysis, statistics, graphing, and reporting, and or getting results out to the tools and the software packages you need in a directly integrated way. And that's kind of the big FCS Express difference. One place to kind of rule them all to help you get that final result. Now, when we talk about final results, everybody has, kind of a different opinion or a different kind of experience with what a final result is. Because for some people, a final result might be a lab presentation that's coming up in 5 minutes and you need to get something out to show to everybody. Might be comparisons between samples, things like fit statistics, comprehensive reports. And for some folks, it might be all of these things, right? And so FCS Express is well suited to handle these, and we're going to go through in about the next half an hour with a kind of subset of kind of results and example types that we think are relevant to the majority of people doing flow cytometry. And again, if you have questions, please make sure to just type into the webinar chat or the Q&A with any questions that you have. We'll be happy to answer those as we go through the call today. And I see one hand up here. I'm just going to lower the hand. If you could just type a question into the Q&A, we'll be happy to answer that or come back to it. So again, one type of presentation, one type of final result is lab presentations, posters, papers, the things that we need to do as scientists to communicate our results to other scientists. And generally, when we're working in other software packages, we're shuffling between flow analysis tools and presentation software, and that takes time and introduces complexity because oftentimes we find customers coming from other tools are working with low resolution graphics and limited formatting capabilities. They have to use other imaging type softwares to, say get things out to 300 DPI because that's what the publisher wants. And that takes time and introduces kind of needless complexity for, what can be made a lot easier using FCS Express. So I'm going to open up FCS Express so you can see we're jumping into FCS Express software right now. And for those of you who may not have seen FCS Express before, well, this is it. And it looks exactly like Microsoft PowerPoint, like Microsoft Office, right? And that's by design because most people who come into Microsoft PowerPoint or most people who come into FCS Express can know that if you wanted to insert a text box, you draw an object on the page, you put text in the box, whatever it is that you wanted to create. You know that, there's probably some sort of text formatting capability, right? You can move objects, you can resize them. And in FCS Express, our objects are also data. There are also, charts. This is our raw data from our flow cytometer. And again, instead of inserting an image, we would come in and say, well, let's insert a density plot. Let's change to our parameters of interest and let's create some sort of gate and click through. FCS Express is telling us, in our software, what you're going to do is drag and drop a gate to open it up and open up a new plot, change parameters, and then we're off and analyzing, and you can see results and charts update in real time as information changes. The other nice thing is we can undo and we can redo, right? So I'm clicking undo, I'm clicking redo, and that will work for every single action that you perform in FCS Express. You're not going to hit the undo button and then something's not going to work like you may have seen in some other software tools. It's going to be really nice and easy to use like that, again, just like PowerPoint. But again, what we're talking about right now is kind of posters and presentations. You need to take your flow data that might look like this in a default sort of plot in FCS Express, and you decide for your presentation. You want a gray background. You want to, kind of put some density to your dots. You want to highlight outliers, right? We can make these changes in FCS Express by right-clicking and choosing Format. And in Format, you can come in and you can make any sort of change that you want for any sort of aspect of the plot, titles, legends, displays, things like that. And it really enables you to again move from kind of the standard display that you get even from FCS Express or any software to something like this where you put a black background. You change the color of the quadrants, you use floating quadrants or these kind of spider looking quadrants instead of standard quadrants. Formatting contour plots to have density scales and counts, formatting histograms. So if you need histogram overlays, you need three-dimensional plots, you need ranges and things on there. We can accommodate all of these changes directly within FCS Express. But FCS Express doesn't really stop there because we know that the last thing that you might need for this is to get this information out, to bring it to your lab presentation, to bring it to, your poster, to bring it to publication. And we enable you either through a one-click option to export to PowerPoint or PDF or pictures of individual plots or pages, or through a batch. And I'm going to show you the batch in a little bit because we don't want you doing a one-off export for, say, 100 data files. But what you're going to be able to see here is that I'm going to choose to, export this PowerPoint presentation directly to my desktop. I'm going to say open it after we're done. And with this, FCS Express is gathering everything that you've done in this analysis, and it has now launched it in PowerPoint, right? So don't be fooled. I know we look exactly like PowerPoint, but this is PowerPoint. We have our objects, we have all of our plots, we have all the different pages from our FCS Express analysis, easily accessible. And the other nice thing is, once you get here, if you're working on a PC, you can actually group and ungroup these objects in PowerPoint. So if you decided you don't like FSCH on there and you wanted to put forward scatter, you don't like the ticks, maybe you just need the distribution of the data to show in your lab presentation. I know this is in PowerPoint, but the way that FCS Express exports the PowerPoint sets you up for success in all that you're doing with your presentations and posters and things like that, right? So keep in mind these tools, and again, keep in mind they don't have to be done one-off like this. We're going to show you in the kind of next example that we're going to go through about how that works. But when we talk about presentations, posters, and papers, the FCS Express way is that we fully integrate analysis and presentation tools directly into one program. Fully formatable text, ultra high resolution graphics. There was no copy and paste that I did. There was no need to use external software for plot enhancements, right? I didn't put a little white text box over anything. And this reduces time and costs and complexity when you're doing flow cytometry data analysis. And it's one of the key tenets that we work with at FCS Express. Now, things become really interesting when we start looking at comparisons between samples, because generally when you're using other tools, you have to shuffle between kind of tables and graphs and copying and pasting, and taking things into Excel and transforming them for Prism, copying and pasting out to PowerPoint to pull it all together, right? I think somebody said they have issues creating figures with statistics on them, right? So with FCS Express, you don't have to do a lot of this. I know other tools have limited calculation tools, limited graphing tools, but when we look at an example in FCS Express, we can make use of integrated spreadsheets, we can make use of integrated graphing, we can make use of direct exports to GraphPad Prism, which is pretty cool. And when we look at an analysis example in FCS Express, you can see I've got a few plots here, different time points, right? I'm not working with one set of data. I've got a 6, 12, and 18-hour time point. I've got some bar graphs down here and pie charts, but this is really cool in that if I move a gate in FCS Express and I have charts that are linked up to that gate, those statistics, the charts, the data, all update in real time through the use of what we call spreadsheets in FCS Express. So, if you're thinking about, or sometimes I think about this when I used to be a grad student, I would show my PI, some sort of analysis, the graphs, and the tool that I was using didn't have integrated graphing like this. We'd say, oh, let's move the gate 1 pixel and see what happens. And then we'd all decide that, nothing happened in the graph. So then we would go back and have to move the gate. And in FCS Express, we can review these results in real time. We can review the gating changes, removing outliers as we're working on those data sets, right? This again kind of goes above and beyond just a very simple graphing analysis like that. I can show you something a little bit more complex where we're actually looking at more replicates, more sets of data. We're calculating p-values, we're calculating levels of significance. We have kind of error bars on these plots. And in this example, again, we have, 2 replicates, 3 replicates at 6 hours, 3 replicates at 12 hours. And on these charts, if I move this gate, you can see like levels of significance, p-values, error bars, everything is updating in real time. Now, I know some critical folks in the audience might be looking at this and saying, well, doesn't this just mean that you can, adjust your gate and get that error bar as small as possible and that level of significance as, great as possible? Well, yeah, you can do that in any tool that you want. This is not for p-value hacking, right? But FCS Express will allow you to come in and say, all right, well, what happens if we remove some of these outliers? Does that impact our results? Does that have an important scientific meaning for what we're doing? And if the answer is no, well, I can hit undo and I can get back to where I was. And if the answer is yes, I can further explore this data and I can, dive in and get the information I need without having to step through copy and paste to Excel, to Prism, and other tools. Now, I do understand though that a lot of people like to work in Prism because it's a really great tool, and they're one of our kind of sister products at Dotmatics, and because of that, we've been able to enable direct exports to GraphPad Prism from FCS Express. Now, earlier I'd mentioned in the previous example that we can utilize what's called batch processing. And batch processing in FCS Express is set up so that if you have, a data list like this, where we have 96 data files, this could be 100, 1,000. The most I've ever seen the data list was 20,000. But you can easily step through all of your data files, kind of like I'm clicking through here to review them. But then I can say in our batch actions, which are here, perform a set of actions like print the analysis, save it to PowerPoint, save a separate PDF page for every sample that I'm working through, and importantly, export to Prism. Or, you can see down here we have export to Excel. And what FCS Express will do is when I'm set up for a batch process, I can come up and I can say run the batch process. So let me make sure I get my data list back to the first iteration here. And I'm going to come over and say run on the batch process. And right now, FCS Express is crunching through the 96 data files. It's exporting results for different gated populations and MFIs out to a GraphPad Prism project. Again, if we were saving PowerPoints and PDFs, it would be doing that as part of the process. And in the end, FCS Express is going to create a Prism file on my desktop, which I got to go over to my other screen here. Let me go pull it up. I'm kind of hidden behind chat windows and question and answer windows, but let's see if I can find my Prism project and pull this up. And what you're going to see is the Prism project that was just created, that I'm pulling over to the screen now, has all of my results for all of my 96 data files. I have them exported into, column tables, XY tables, and Prism can also be set up so that I can template this so that if data hits this Prism file, you can see that I already have my drug treatments grouped together. I have things like the dose response grouped together, and all of this is being automatically charted through the use of these templates in GraphPad Prism. So again, I think this is a huge time savings. And again, what we've looked at in FCS Express is that we use integrated spreadsheets, integrated graphing tools, real-time data update capabilities to help you get results, to help you compare between samples. And again, with that direct export to GraphPad Prism, that can save a lot of time. And that's going to save time. Time is money. That saves costs. We are saving complexity because we're not working through various different tools. And because there's no copy and paste, that is importantly reducing the opportunity for errors. I guarantee if you're copying and pasting over and over again every day, someday you're going to make a mistake. We're all humans, right? And FCS Express is designed to kind of overcome that with the efficiencies that we offer in the software. And another kind of really great example, there's a lot of people out there who are working with IC50s, they're working with titrations, QMB calibrations, and they're doing fit statistics. And if you're doing that in other software, you have limited or no calculation tools, right? You have to copy and paste to Excel, you have to go out to Prism. You have to graph that data and then export it to PowerPoint as well. And it's really an inefficient use of time because simple gate changes require a total repeat of the process. It's really hard to batch or automate and repeat that analysis. And then you also have that increasing complexity of archiving, CSV files, Excel files, Prism files, PowerPoint data, FCS files, analysis templates, right? And FCS Express, really helps overcome that. And I'm going to show you in kind of two examples here. One where we look at an antibody titration that's going to pop up here, and I should mention, if you aren't doing antibody titrations, you should, right? They're going to help your science and your experiments get better. They're also going to help you save some money. But we understand with other software tools, it can be challenging and complex to do this, right? And when we work in FCS Express, we can work on multiple pages in a layout. So on this page, we have a 7-point series of data files that are being plotted and displayed. And on the front page, we have our final result, and we have a curve fitted graph. We have these spreadsheets, right, that I talked about in FCS Express that work like Excel, right? If I enter in a value and I say, give me a formula for the standard deviation of, say a few cells in here, it's going to report that value because we work exactly like Microsoft Excel for these spreadsheets. And these spreadsheets and these graphs and these charts, they all interact. So as I move this gate, it's updating everything on our curve here for titration. It would be updating our ICEC 50 curves, right? And you probably see that little green point floating around. You're probably wondering what that is. Well, that's our final result. This is an antibody titration. And what we want to get to is the amount of antibody to use for a specific clone or a specific titration point that we're using, right? And again, you can use this analysis template as a template. We can use this over and over again. So if I have another series of data, I have another set of 7. I click in the data list, it switches over to the next set of 7 data files, updated everything on our charts on the second page, and that final result for this clone is reported. And we're even doing something a little fancy here where we use metadata or keywords from the data file to report the name of the clone. So any sort of metadata or information you're storing with FCS files can be pulled into your analysis and pulled into part of your report. So again, working with, these sort of fit statistics becomes very quick and easy to do in FCS Express. Um, and I should also mention that with FCS Express, we have a number of antibody titration templates, things that you can use as your own kind of starting point that are meant to be universally applied to any data files. So again, when we work with things like fit statistics in FCS Express, we don't have to rely on copy and paste out to Excel, out to GraphPad Prism. I love Prism. Prism's a really big part of what we do at FCS Express and work with Dotmatics. But if we can reduce the number of steps and complexities to get to a final result, it can be easier. And in other software tools, you just don't have that capability. So again, with the integrated curve fitting, the ability to batch and reuse layouts and templates in FCS Express, you're going to reduce the time, you're going to reduce the cost, the complexity, and the opportunities for errors that come in when you're having to copy and paste and move between lots of different software tools. So, in the interest of time today, I can't go through all of the other specialized analysis in FCS Express, right? We have the ability to work with heat maps and high content screens. We can work with large data sets very quickly. We can work with multi-plots and multi-plot compensation, spectral data unmixing, R and Python, high dimensional data reductions that are natively built into the software, so you don't have to know how to code for R and Python. You can work with things like UMAP and TISNI and SPADE and other clustering tools. You can work with image cytometry data sets and cell cycle and modeling sort of things in FCS Express. Again, this has been developed over 25 years of working with scientists and working in the industry to provide these tools. And I think as the last example to kind of show you how things really kind of come together for folks in the end is with comprehensive reports. Because we know with other software tools, again, you're having the copy and paste into Excel and Word and PowerPoint and generating PDFs from that. It can be difficult to automate, to batch, or to repeat that analysis. So, kind of the idea of generating a report where you have the most important statistical information presented up front and be able to crunch through that report for as many samples as you have can be challenging with some of the other software tools on the market. So in FCS Express, again, we build this sort of functionality into the software. And I'm going to show that to you with kind of two more complex type of analysis. One is what we would call multi-tube panel, which a lot of folks who are doing clinical analysis, working in conventional cytometers, they have to include multiple tubes to achieve something like a 20 or 30 marker data set. Whereas, people who are working with spectral cytometry, which I'll show you in a minute, can achieve that with one tube. But the important thing about what I'm showing you here is that we have used a template. And this template has pulled in multiple tubes that make up, part of, probably like a 20 color or 20 marker analysis here. And we're summarizing all of the information from this analysis on the front page. And all of this is being done through page tabs in FCS Express, where we're analyzing viability, we're analyzing B cells, we're analyzing, myeloid markers, right? And again, the big efficiencies that come with this is that you can set this up and you can crunch through it over and over again using the batch process. But these statistics, everything that's reported here, these are not static one-time statistics. As I move something like my lymphocytes gate, you're going to see that these values and these numbers are updated in real time. And we also have some unique functionality going on in here. Whereas, if I move this lymphocytes gate out to a certain portion, we're flagging this result in blue because it falls outside of some sort of expected range that you as a researcher would set up. We also have the ability to assess, not just number values, but we can assess and report textual values. So in this viability assessment, it says good. But if I came here and I started looking at a two-page analysis and I come in where my viability is being set, if I move this marker, you can see it's moving to unacceptable. Over here on the left, it says good, suboptimal, unacceptable. So we're getting information that's based off our results and our data files being reported automatically, both in text, numerical, and flagging in different sorts of ways within the software, which becomes invaluable for QC teams and people looking for efficiencies and crunching through many hundreds of data files and identifying the ones where there may or may not be issues. But I think another kind of nice example of working with these comprehensive reports is if we look at something with, say a more modern sort of cytometer, where we're looking at, a spectral data set in the context of, in this case, I'm going to show you an example of omic 69, which is a omip which is looking at 35 to 40 markers and how that's being reported and analyzed in FCS Express. And in this layout, what you're going to see is that we have, again, lots of different pages. We've even broken down all of the reagents being used here. This information could come from metadata from your FCS files. We're breaking down with kind of displays that are going to be really helpful for communicating these results with images and this individual statistics summary page, right? Where we've broken down for all the different subtypes that we're interested in, what those results are and what they represent. And again, all of those statistics are coming from multiple pages in our analysis where we've quickly and easily, broken down the different gates, the different, subsets of populations that we want to use and we want to look at. And at any time, we can break this down into multiple page views. So if we wanted to, look on one side of our screen at something like the, statistical report page here, I can scroll back over, not initial gating, right? But we can look at our statistics summary. And if we do something like update our CD8 gate, what we're going to see is that all over on our statistics page, information is updating in real time. And again, you might be wondering, oh man, that's a lot of information, a lot of statistics to be looking at. How is that going to be useful to me on just like a PDF report, right? Well, keep in mind that through the batch actions, we could export that to PDF, but we could also export it to Prism. We could export to Excel, and we can also add customized reports in here to directly integrate with lab information systems, lab management systems, and even tools from Dotmatics like Luma, which are designed to intake large volumes of kind of statistics and metadata and sort and get that organized in a way for you to take action on, not just for one experiment, but for all of the experiments that you're working on, right? So again, when we're working in FCS Express and we have these kind of comprehensive reports, we have these sophisticated page layout tools that help you work between, all the different analysis and statistics you need. You've seen the real-time data updates that are working with very large data sets. I'm not on some supercomputer here in my office. I'm on a machine that, it's probably very typical of what you might be using as a researcher. I'm getting really great speed and efficiencies from this using FCS Express. We can bring those statistics and reportables, kind of custom calculated fields all together. And those report elements can be flags. They don't have to be simply numbers. They can be textual flags, font formatting, anything that you want to help again reduce the time. If time is money, the cost, the complexity, and the kind of opportunities for errors that might creep in with, that sort of thing. So when we look at FCS Express as a whole, and again, as this kind of high level overview of the software, when you look at what you're doing and other tools, which are kind of over here on the left, the analysis portion is actually probably a minor part of the process because, graph generation reporting, it takes as much time to generate graphs, to generate reports, as it does to complete the analysis in other tools. But if we take that and we move that over to FCS Express, what we expect you to see is that things like graph generation, reporting, getting statistics aligned with data, the actual analysis by using batch processing, using our efficiencies, those time portions really, really shrink using FCS Express, right? So I should put a disclaimer here, results can be variable, but what that opens up is a huge amount of time savings. And we've seen this with customers time and time again. When they make that switch and they come over or they start with FCS Express, they're able to recognize a huge amount of time savings with their analysis and their reporting. So what does that mean for you, right? The way I think about it is, how do you want to be spending your time? We have kind of researchers over here on the right, who are kind of fighting with putting little white text boxes over access labels. They've been copying, pasting all day. They've been working between 5 different software tools to generate a report. And we have kind of people over on the left here, right? They're publishing, they're getting ready to go into presentations, they're getting back on the lab bench because FCS Express is enabling them through the efficiencies and the design that we have in the software to really help move them along. So, we really see FCS Express as a time and money saver because of the ultra fast computation that's built into the software, having the ability to batch and repeat through reporting. You can build that template once and reuse it over and over again, and you have the ability to format and create the high resolution graphics very quickly and easily. And again, for really any researchers, but folks who are in the quality space, folks who are working in regulated environments, the ability to reduce errors and complexities by, limiting copying and paste, by, having faster training even because you're wearing one software package and you're learning something that's very similar to Microsoft Office and PowerPoint and Excel, right? You can always kind of refer back to what you would do in those softwares to help move you ahead with FCS Express. And again, by having kind of single file archiving where an FCS Express layout can contain the raw data, it can contain your plots, your statistics, your graphs, and that final report, that really helps from a QC kind of procedure as well. So, I know we'll be following up with everybody. We'll give you a link to our free trial, which is good for 30 days. It's fully functional, so you can load any of your own data in there. Our team will be following up as well. Um, so if you wanted to arrange for any sort of kind of one-on-one trainings, any sort of Q&A sessions, keep in mind that we have that world-class product support. That's really a key differentiator between FCS Express and other software tools that are on the market. There's a number of kind of webinars that we have coming up. We have web-based trainings, and we also love coming out and visiting customers in person. So, if you think you're the kind of site where, we'd be able to come out and, speak to the crowd and speak to some of the folks in your institution, we'd be happy to try and put that together. I had mentioned earlier, we have our technical specialists with, a wide-ranging set of expertise and time zones for virtual and in-person training. And I really am proud to say, I think FCS Express customer support is second to none. And I really encourage you to check that out. Email us at support@denovosoftware.com and see how fast you get a reply. So with that, I'd like to say thank you for joining us today for about the last 45 minutes to kind of again go over some of the high level features and functionalities with FCS Express. Getting an overview of what are the tools and efficiencies that can be realized with the software. And if you have questions and answers, make sure, or I'm sorry, not answers, but if you have questions, make sure to put your questions into the Q&A chat and we can review them. All right, and I see we do have a few questions coming through here, and so it looks like one question is around do we offer essentially data analysis services for making templates and reports for customers that purchase our software? So we don't offer, bespoke, analysis, kind of template building outside of our kind of standard training services. Or generally, if a company or institution comes online, they may hire us to come out and do some in-person or virtual training. Some of that can be included as part of that, but we don't have a kind of a one-off bespoke, create a template type of service. However, if you go to our website and you look at our kind of applications examples, you will notice that we have a number of kind of templates that we've set up to be as universal as possible. And that means that no matter what instrument you're coming from, what kind of markers you're working with, we've tried to design some templates to help you get off and running with results quickly and easily. But then again, we also have a lot of confidence that folks are able to create and work in FCS Express very quickly and efficiently on their own, right? So, we have a lot of videos, we have a lot of tutorials, example data that you can work from to get off and running. Great, and I see there's another question asking about how does FCS Express dovetail with tools like OMIC, and I think that kind of more generally applies to the tools at Dotmatics because we have a lot of great software offerings, in the flow cytometry world. Dotmatics is the parent company of OMIC. It's the parent company of EasyPanel, right? So we work very closely with the team at OMIC. OMIC works very closely with our team. There's a lot of kind of similarities between products, some differences in the ways that the workflows are kind of defined. So I always encourage folks, if you're looking for the difference between some of the different Dotmatics software offerings that are out there, free trials are offered and you can go and grab them and our team would be happy to work through you with either one. Easy panel panel design tool, we do have direct integrations with FCS Express, so if you're doing spectral and mixing with FCS Express, you can actually export directly to EasyPanel to check against their known set of reference spectra to validate, how close those are being, or how close those your unmixed results or your real results align with known spectra. And a similar tool is offered through OMIC. And I'd also say, we have direct GraphPad integrations from, many of the Dotmatics tools that are out there. So it's probably a little bit of a long-winded answer to the question, but we have a lot of kind of integrations between Dotmatics products that I think everybody can benefit from. All right. So I don't see other questions in here that haven't been answered already as we kind of went through today. So again, if you have questions after the call, make sure to reach out to support at denovosoftware.com. Make sure to visit our website for denovosoftware.com if you'd like to go grab a free trial. And again, we really appreciate your time coming out here today to learn more about the software. I'm going to answer one more question. I saw one come through the chat last second here. It says, is there a tutorial to learn how to organize data to export into Prism? Yes, we have web resources, we have videos, we have written instructions, tutorials. So, I encourage you to go to the De Novo Software website, type in Prism in the search, and a lot of those materials are going to come together for you. So again, thanks for everybody's time today. I really appreciate it. And we'll be in touch, and I hope to speak to folks again soon at some point. Have a good rest of your day and rest of your week. Take care. Bye bye.